We're going to speak about confined cell monolayers. And I swore a long time ago that I would never use this slide again. It's, used usually to introduce the difference between individual behavior and collective behavior, in the sense that we've been done in my group in the lab for a long time now, several years. And the idea behind it is that to understand these shapes that, in that case, starlings, birds met together, you have to understand the interaction they develop between them. It's pretty obvious, but these interactions give rise to the emergence of this phenomenon. And you have the same thing in fish. It's maybe even more well known. Fish schools have been, the subject of many studies in the past. And structures like this, where fish swim in circles, have been the highlights of many Discovery Channel programs, let's say. However, one of the things that struck me recently, and the reason why specifically I put back this slide today, is that most of the time, these images are taken in aquariums, fish tanks. And fish tanks, as you know, have walls around them, most of them circular, right? You see where I'm coming to. It's actually a structure that emerges not only from the collective behavior of these fish, but also from the boundary condition they encounter. Of course, for birds, sky is the limit, as they say, and you don't have the same sort of effect. But here, the boundary condition might be very important. And of course, that's what we're going to discuss today, the influence of the system size on the behavior, on the collective behavior of the cells. Why is it important? Well, it's pretty obvious, actually. It's important because in vivo, a lot of phenomena happen at these scales where small tissues are de facto confined in small areas. This can happen in morphogenesis, in embryonic morphogenesis, where by definition everything is restricted and small, and where some colonies of cells have to migrate, for instance, from one place to another to fulfill what they're going to become later. In other situations, like cancer, you start with a small population of cells that are well defined and with clear boundaries again, and that's going to develop its own collective behavior before the dissemination of the individual cells in the organism, and then another story begins. But it's also important in biotechnology. Cell arrays have been around for a few years now, and they're basically small chambers where there are a relatively small number of cells in, and what you can treat in parallel with drugs, siRNA, whatnot. But obviously, in these situations as well, the space is limited, and the cells are going to react in this limited space within these constraints, these spatial constraints. And maybe, well, we're going to see that this can have a big influence on the behavior of these cells. The other question we can ask ourselves is, we know sort of how a single cell behaves once it's on the surface, and that's by protruding a lamellipodium at the front, retracting at the back. However, it has been the issue to transpose this picture to many cells, to population of cells, is a different story. And that leads us to what is called contact inhibition. Contact inhibition has two meanings, actually, in biology or biophysics. Or it can be interpreted as contact inhibition of locomotion. And that was actually the first time this term was coined by the Abercrombie group in the 50s, and then the whole British school that came afterward. They used this term, contact inhibition of locomotion. And basically what this tells you, this is the original phrase that you can find in the paper, but basically what this tells you is that if two cells encounter one the other, they can't continue like this. They will go away one from the other. Okay, this is good, and it's probably true for single cells, but how can you transpose that to many cells? Of course, many cells that interact together, that have adherent junctions, for instance, between them, They can't migrate as a single cell does. They have to take into account all the others. But when there are cells everywhere, when you deal with a confluent monolayer, if the Abercrombie sentence was right, then everything should just freeze, stop. Because wherever they go, they would encounter another cell, and then they would just don't go anywhere, which is obviously not the case. This is a monolayer of the MDCK cells, some of these epithelial cells I was talking about that develop the cell-cell interaction, adhesive interaction. And you see there is a lot of movement in there. So contact inhibition of locomotion already is not very, very clear and not very well defined, at least at the scale of the tissues. We know how this works. The cell still develop a lamellipodium. The picture that I just told you was not quite right. They could develop lamellipodium that go under the cells around them, and then they can still manage to migrate, even if they touch each other and they develop adhesive interactions. But there is another contact inhibition. And this other contact inhibition is, and for many people, that the only definition is contact inhibition of proliferation. When cells, the tissue get confluent, cells just stop to divide, they become quiescent, and everything is done and that maybe it can be one of the hallmark of cancer. It's monolayer of normal cells would do that, and monolayers of cancer cells would develop cells in the third dimension and get structure, etc. This is a cartoon, right? So we'll come back to that at the end. This, again, has triggered a lot of studies which, and the same name infuriated the Abercrombie and Al group because they didn't want the two notions to be misinterpreted as a single one. And indeed, this is not the case if you take this movie of a spreading monolayer that I took from this reference. You see that it can spread, develop a colony, but then it's very heterogeneous in terms of cell size. And depending on where you're looking at and at the time at which you look at, cells that have a small area will make huge amount of time to divide. Put it that way. That division time will increase as the area of the cell decreases. The more dense you are, the longer it takes to divide. And that's a way to express contact inhibition, except that it's not, just a clear threshold. It's just a continuous process. Okay. So in any case, well, some people have tried to go around this density-dependent inhibition. Okay, all right. The thing is, we need tools to study even the simplest level of what I just described, these two aspects that I just described. We need control condition to study cells after they get to confluence. And for us, these control conditions are micropatterning. So we micropattern cells pretty much as Mathieu has done as a single cell level, only we do it at many cell level. So you can see here our patches where a cell can adhere are 250 micron in that case. It can go, let's say, from 50 to a millimeter. 50 micron to millimeter. The thing is, it's a population of cells we're studying, not just one. And of course, if you go too big, then you go back to a monolayer situation, which is not what you want necessarily. The other good thing is that we have the chemistry tool to develop an anti-fouling treatment here that I won't detail, just to say that it's very robust. Cells don't go there, even for months. We tried that. So cells are really confined where they are. And the other thing, of course, is with any microtechnology tool, we can make many of these patterns on the same slide and study them in parallel, get good statistics, etc., on a single experiment. It's actually easier to do than trying to have the resolution of a single cell patterning. So what's going on? If we plate cells on such a substrate, which is non-adhesive here and adhesive there, what's going to happen? So the cells develop on their pattern, as you expect. They reach confluence by division. And then you see they develop some motion. And at first, they wiggle around a lot. And then you see we are coming at 30 hours here. It becomes much more quiet. The movements are much more localized. And then after very long times, you can see a structure appearing at the rim of this patch of cells. So let's do that again. It becomes very quiet, and then you see this structure here. And let's see again the beginning maybe. So you see they wiggle around, they turn, they go in all directions. A lot of movement, as I said. Confluent doesn't mean stop movement. Only high density do. The cell density, of course, increases even after confluence because cells divide, and they increase their density on the patches by a small amount that is very repeatable from experiment to experiment and from patch size to patch size. How do they get this stable cell density on the patch? That we come back to that later. Just to say that we recover the curve that I showed before. The division time as a function of the area of the cell increases sharply when the cell becomes too small. That was expected. That's what you have on unbounded surfaces. That's nice and well. What we can do more was to monitor the displacement of the cells within these individual patches, and we do that by using PIV, which allows us to map all these displacements without having to track single cells independently. So when we do that, what we observe is that the velocity decreases. That's what we see with our eyes. The cells, the movement of the cell progressively freeze, and they come to almost a stop at the end. But what we see also is that they do that with huge fluctuations during this slowdown of the velocities. And so we're trying to explain that. That happened at the first 30 hours is here, and we said that after 30 hours, everything gets pretty much quiet. So let's focus on these first 30 hours, and we have the velocity on each point, because we have all the displacement mapped, so we have the velocity, we just have the time interval. So we can decompose the velocity we obtain at each point in the orthoradial and radial direction. Let's do that, decompose these velocities on each of these directions. So the orthoradial, let's start with that one here. The orthoradial velocity just tell us how much we rotate on each pattern, right? We have all the cells here, and what we can see is that we do rotate because we have a succession of positive and negative velocities, so we have rotation in both directions. And the radial direction of the velocity tells us that it's more complicated than that. Here, if we project all the velocity on the radial direction, we see that we have a succession of colors that we're going to discuss a bit more later. And after 30 hours, everything just dies out, pretty quiet. So that's the orthoradial again. What we see is that depending on the size of the patch, we have always a maximum of the velocity at the edge of the domain. So that's the chymograph giving, I'm sorry, I should have precise that. That's the center of the patch, that's the border of the patch. Everything has been integrated over all angles, right? And that's the time. 30 hours is our limit. And what this tells us is that basically the patch turns in one direction, that's the red, and then in the other direction, that's the blue. It stochastically changes direction. If we try to analyze the time at which it changes direction, there is no real period there. And it tells us also that it's maximum at the edge, so it rotates as a solid body, basically, right? So it's not much surprise here. That's our fish from the beginning, or goldfish you put in a fish tank. They were going to rotate, right? Return. Okay. So that's sort of expected. What was not expected at all was that here, the maximum of the velocity, so that the radial projection, I'm sorry, again, that's the center of the domain, that's the edge, and that's the time. And what you see is that you have maximum signal, either red or blue, at the center of this domain here. It is at mid-radius. That's where you have the maximum of signal. And when you check exactly what's going on, you see that you have a succession of colors, red, blue, etc. So basically what you have is a breathing oscillation. You go in one direction, and then you go back, and then you go again. So radially, there are displacements that are described by this breathing, if you want, and which is maximum at mid-radius. So if we project all this over a single point, if we project all the radii over a single point, we get that's the velocity versus time, and indeed it oscillates between values before dying out again, as we said. And is there something more interesting than in the stochastic changes of direction we have in the autoradial direction? Yes, it is. If we now make the Fourier transform of these breathing oscillation, if you want, you get a frequency that actually appears to scale with the radius of the domain. I say appears to because we're going to see another explanation just in a minute. But basically, it depends on the radius. We have these breathing frequencies that depend on the radius, something like the soft mode of physics state, solid state physics. This phenomenon doesn't depend on division. If we put that drug, mitomycin, it doesn't affect the period of oscillation, but it is very much linked to the collective behavior, of course. If we put a drug that, a growth factor that just destroys these interactions, or low calcium that disrupts cell-cell interaction, physical interaction, a direct interaction, then we lose that pretty much. So how can we understand that? And for that, we collaborated with Vincent Hakim at the École Normale, and Vincent had this very nice model around for some time. So let me describe it to you. It's a cell-based model, and we enter the interaction between cells. That's the adhesion, if you want. We enter the mobility in terms of a noise that cells are, actively moving around. And this is a dissipation, right? That friction, adhesion to the substrate, adhesion between cells, viscosity, whatever. This term is the most important one. That's the coordination. That's for a single cell surrounded by its neighbors, is going to take the velocity of the neighbors, the average velocity of the neighbors with a certain weight. Beta gives its weight. So every cell, that's called a V-check model, every cell takes the average of its neighbors with a certain metric. I don't get into the details. So why were we trying to use this model? It's because we know from previous experiments that it works very well to describe collective cell migration. And so I don't want to go into the details here, but we studied collective migration for some time, a lot of time. And what we could get is that this interaction and this alignment term gives rise to very correlated flows within the monolayer as they migrate on free surface. That's the main difference with what we have now. We have free surface on which cells can migrate. And if we map the displacement of these cells by the same technique, we see that they show a velocity-velocity correlation length, which is about 10 cell diameter. And this is very well accounted for by the model I just showed, by this V-check model. The experiments are here, simulations are there, and basically we can describe these flows, these swirls, these correlated flows with this model very well. So the idea we had was, if it works that well for migration, does it work as well for cells still within their adhesive patches? They don't migrate anywhere, they just migrate within the patch. See, it's easy on a computer just to set the boundary condition, and Vincent did just that. And what you can see on the radial component of the velocity is that you recover this red, blue, well, it was blue in the other picture, this succession of colors, showing that there are indeed oscillations in the movement. So the simple model we had that worked very well for migration works also in confined monolayers. And then if we compare quantitatively, because we have determined the parameters in the previous migration experiments, so we don't fit anything, we just plug the same parameters, and what we get is a quantitative agreement with that. So it really appears that these breathing modes are just due to the boundary condition excited by the stochastic modes of each cells. So that works well, but what about the long times? We saw that this was a different story, and here is what happened at 60 hours, so much later. What you see here is a lot of white stuff. Doesn't appear very clean, doesn't seem very nice. We try to get better pictures usually. But these are actually the result of cells that extrude from the monolayer when the monolayer gets too dense. And that's, you have a confocal image here, a very nice one, that shows exactly that. When cells get too dense, before they could trigger any complicated pathway or mechanism just to regulate the cell division, they begin to extrude cells from the monolayer. It's a very good, very fast quality control, population control. It's very, well, okay. It's very easy and very efficient. And so all the cells here, the result of division, the issue whether they're still alive or dead is still an open issue, but they don't participate to the monolayer. And so this extrusion works very well until a certain point where you see that at very long times there is a development of this, what we call a rim, that you can see in confocal. And that's really the pluricellular structure that emerges from this bidimensional monolayer. Here it is. And I mean, it can be huge. It can be a very large thing. It's only, well, mostly due to the presence of a border and nothing more. Although sometimes you can see it also in the middle of these monolayers. But you can see it systematically at the edge. It doesn't depend this time on the radius of the border, of the domain. It's really an edge effect. And if you take two small domains, what they do is that they make this little droplet after some time. So they really go three-dimensional, which is a surprise to everyone who has studied MDCK cells. They're a prototype of epithelial cells. They stay in 2D, I mean, in every textbook. Actually, they don't. If you give them the availability to grow in the third dimension, they do that. And they do that by proliferation. If we compare the growth of this structure with the division time, should be here, one of these, what we see is that the two are very compatible. It's not a flow of cells from the interior to the exterior. It's really due to proliferation, and it's a differential extrusion. That's how we call it. This is our domain. We measure the number of cells popping out individually from the center and from the edge. And already visually, you can see that there is not much of this individual extrusion on the edge, right? This is where we have the rim. Cells haven't extruded individually. What they have done is that they have extruded individually in the center, and then we lose them. Oula. and then here they have extruded collectively. They make this rim, this 3D structure, which is a very interesting, so that just recapitulate that. In the center, they extrude individually. At the edge, they extrude collectively, if you want. They still develop cell-cell interactions, but without the interaction with the substrate, which is still not clear how they can do that, because usually the common picture is that they can't do that, but they do. Okay, so, and that may be, yeah, I should precise that should maybe the precursor step of the formation of an organ. Because these cells are coming from kidney, they make tubules in the organism, and making this 3D structure is already a first step toward one of these kidney tubules. So in 2 minutes, I'm going to show you a different type of cells, which we call nematic cells. And these are fibroblasts. They're very elongated, and when they move alone, individually, they don't have a preferential sense of division, direction of division, right? They go back and forth. They're basically apolar if you look at them with a sufficient time scale. When we put them together, many of them, they interact physically, just by steric interaction in a way. And what you can see is that they make these very nice domains, very well oriented. I hope you can see that. And so this looks like a nematic. So we went a bit further. These are not the best pictures. Here we code the orientation with the color. And you see that the defined domain with even colors, meaning the orientation within each domain is very good. But there is a succession of all these domains. So I'm going a bit fast here. If we define an order parameter that precisely describe how well we are oriented over the field of view, we see that it saturates, not because the order is not very good at a local scale, but because the field of view is full of different clusters of cells oriented differently. The size of these domains is a few hundred microns. We can determine it better with measuring the correlation function. Is it a nematic? I used the word already. And our conclusion is yes, it is. The proof is in the defect. not in the pudding. And you can see the two characteristic topological defects of the nematics. It's a clear evidence that this is a nematic state, fulfilled by these cells. We saw that if we use a very large scale monolayer, we have a juxtaposition of these different patches. And the reason why is that as the cell divide, the density increases, the order increases also because you force them next to next. But also, the displacement freeze in this, we can use a jamming transition to explain that. They basically stop moving after some time, as we had in the first example. So the two act antagonistically, if you want. The more you go, the more they orient, but the less they can displace, move to get this orientation. How can we get from that situation a perfectly oriented monolayer? Well, we can take our lessons from classical nematics. All right, that was a question. So I give the answer because we don't have much time. We can use boundaries. That's what people do in nematics. Not I'm an expert of it, but that's what they do. If you use two plates and you put a nematic in there, you orient them just by the boundary condition. Can we have the same sort of effect here? We can actually, because we can plate the cells in stripes. And what you can see is that eventually, you can see it here already, they will come to a perfect order within these stripes, provided they're less wide than this correlation length we had before, which was around 500 microns. So you can see here it's pretty impressive. They're all oriented in that direction, all of them. And the other parameter goes to 1, basically. I'll skip that one. So that happened because, as in classical liquid crystals, cells align on the edge with the border. They get perfectly aligned here. So again, that's the histogram, that's the orientation. The color code is the orientation, and that's the width of the stripe. Time goes that way. So you orient very well. You read already at the edge. But as your density increases, this order goes toward the center until the whole stripe is fully oriented. That should be a movie too. So you're going to see that. You red at the edge, and then this red color propagates toward the center, as you can see here, until you're uniformly red. You're uniformly oriented in that direction of the. To end, that's probably my last, one before last slide. We can have two orientations compete one with the other. So we make these cross designs. The correlation length is probably that one. And so I won't comment all of them, but basically if you below the correlation length, which is the case here and there, here you have more cells, so they get an orientation that is undisturbed all along the length of this arm of the cross, whereas this one, which is smaller but has less cells, get interrupted here and there. If you have two 200 micron width arms, then they compete and one of them wins. Not always the vertical one. It can be the other one. One time over 2. And then if you have more cells in that direction, then it wins. And then if it's too wide already, OK. Then, OK, my time is up, I guess. I just want to thank everyone. Whoops. This is weird. OK, here it is. OK. So I just want to thank the whole group, of course, and the two students. Maxime Desforêts is the one who did the first story on the confined patches, and Guillaume Duclos is here. Is the one who did the cellular nematics. And we were up with all these people and all the members of the groups. And OK, you can read the last line of yourself. Thank you very much.
OK, thank you very much. Now we have time for a couple of questions. So I have a first question to start. What happens in cell? Does volume change because your density?
So that's an open question. I don't know. There are many different and contradictory reports over that. I think the volume changes. I don't know by which amount. You have to keep in mind that this is a compressible medium in the sense that they get denser, but they get higher also. Yeah, sure. That's what you meant. Whether they lose volume or not, I'm not sure.
Other question? Okay, Mathieu, please.
Larger tissues and then detach them from their adhesive substrate? Or to have floating monolayers? Because here each cell is bound to the bottom somehow.
Yes. Well, as you know, some groups have done that. Maybe.
Detaching is not easy.
Detaching is not easy. While keeping them together, of course. Yeah, that's not easy. We haven't tried. It should be possible, but we haven't tried.
Yeah, it would be interesting to see if you have some stress stored in there.
Yeah, of course.
And another question is, did you check I don't know if you image the mitotic cells in there, and you check the mitotic cell, dividing cells. Do they have an oriented division also in these very oriented fibroblasts?
So in the fibroblasts, we haven't checked. We have checked in the other configuration, and there it's so dense that it becomes difficult to exactly see. Cells are very small, and it becomes hard to see exactly what's going on at division. We have the two daughter cells, and we know where they go. And that's how we know that it's the rim we have at the end is due to this cell division. We can track them. On the fibroblasts, we haven't checked. My bet is that they divide along their axis, as you expect, but we haven't.
And the shapes, are they spherical? As they get denser, do they get more spherical?
The fibroblasts, that's what.
Or any of as a layer get denser, do you get?
The other one, yes, they're still pretty round, and as you expect. The fibroblasts we haven't checked because they keep some orientation in that case. So I don't know. I don't know about that. Maybe you know.
They're still elongated.
They're still elongated.
Any other question? Yeah, please.
Maybe I did not follow. So what determines the width of the ring?
What determines the width of the ring? That's a very good question. We're not sure. It's really an edge effect, and it propagates over a certain length that we think is typical. I mean, it's 50 microns or so. And this is sort of compatible with the physiological diameters you encounter in tubes, in kidney, for instance, in other structures. Although there is a lot of dispersion there. So we're not sure. We think that's related to this idea that it's a morphogenetic step, and that's a response of the cells per se to the confinement. But I mean, that's saying I don't know. It's close.
Okay, thank you.
Thank you very much.
Oh, sorry. Last.
No, it's a very naive question because it's not my field. You were mentioning that the effect that takes place at the ring has some connection or potential connection with tissue formation in vivo, I would say, or in is this connection strong enough to think that we can use this effect to make organs?
So that's a bit the idea, yeah. We like to think so. Of course, I don't have evidence of that. I'm not sure, but it's a possibility. If it's really, you know, to trigger the formation of a real kidney tubule, you need growth factors, 3D environment, a lot of things. But what we show is that confinement itself is already enough to trigger at least the transition from 2D to 3D at the tissue scale. So yeah, that's a step maybe in that direction.
Okay.
Okay, thanks.